Let's go ahead with our next speaker, who is Dr. Petra Bacher from the University of Kiel. Her presentation is entitled IL-17 Immunity from Aspergillus to Candida.
So, thank you very much to the organizers, first of all, for the possibility to present some of our data here today. So I'm going to talk about Th17 cells, actually, and also how our immune system maybe sometimes mix up things that may result in pathology in the end. So T cells can be regarded as central organizers of immune protection, but also of immune pathology. And these T cells, they have some characteristic features, and the most important one is that they have an antigen-specific T cell receptor, so they can highly specifically recognize and react against a certain antigen. And once they're activated, the T cell response is not uniform, but we have functional differentiation, meaning that we have different T cell subsets that all elicit different effector cytokines, and in this way provide protection to the different pathogens that we encounter. But of course, if the T cell reaction is somehow inappropriate or uncontrolled, it can also contribute to pathology, like, for example, the development of autoimmunity, allergy, or inflammatory diseases. So the important point here is really that the type of the T cell reaction that is generated in response to a certain antigen, this plays a major role for the decision between protective immunity or immune pathology. Now, one very famous T cell subset that is associated for a long time with fungi are actually these Th17 cells. And Th17 cells, they are in general regarded protective against different fungal pathogens, but we all know that fungi are highly diverse. So for example, we have Candida albicans, which is mainly a gut commensal, where Th17 cells in the gut are also considered protective. But of course, we also have other fungi, like, for example, Aspergillus, which is mainly inhaled into the lung. And increased Th17 responses in the lung are more associated with pathology, for example, in patients with asthma, COPD, or cystic fibrosis. So our question here was whether fungus-specific Th17 cells might contribute to this pulmonary inflammation in these patients. And now to address this question, what we wanted to do is to really look at the fungus-specific T cells directly in humans, because we think that these cells carry, of course, important information on the type of the T cell reaction that has been generated, and also on the host interaction status with these different antigens. But the problem is somehow illustrated here. So T cells that are specific for one particular pathogen are typically quite rare within a number of T cells with other specificities. So the question is how to directly detect these CD4 T cells that are specific for certain fungi. And to do so, we have developed this assay that we call antigen-reactive T cell enrichment. And in this assay, we simply take human blood or tissue samples. We can stimulate these cells with fungal lysates or proteins or peptides of the fungi. And after 7 hours, we analyze the expression of the so-called activation marker CD154. And this marker is only expressed on those cells that have been activated via the T cell receptor. And now in reality, it looks like this. So if you stimulate healthy donors, for example, with Aspergillus lysate, we get a small induction of this marker of the 154 positive cells. So each dot here is one cell. So if we stimulate or analyze like 1 million cells, we get around 100 of this Aspergillus-specific T cells, so indicating that these cells are really quite rare. And therefore, what we do or added to the system is a magnetic enrichment step, so we can really filter out these few cells from larger input cell numbers. And this really results in a much bigger population of these antigen-specific cells, which we can characterize, of course, with much higher precision. So we use now the system to first look at the T-cell response against two fungi, so Candida albicans and Aspergillus fumigatus, and we used whole lysates to stimulate the cells here. And if you do this for Candida and look now on the cells that have reacted to the fungus, we see somehow the expected pattern. So these cells, they produce a high amount of Th17 cytokines, IL-17, and also IL-22, and we also find a little bit of interferon gamma production. But interestingly, if we do the same experiment with Aspergillus, we find only very few IL-17, IL-22 producing cells. And this pattern is actually true in many donors that we analyze. So always against Candida, we get a clear Th17 response with production of IL-17, also IL-22. But against Aspergillus, these Th17 cells seem to be rather low, and typically below 10% of the response makes Th17 cells. So we thought maybe there's something about Aspergillus, that it does not generate this type of response here. And so we went on and actually screened a larger panel of different fungal species here. And again, looked for the Th17 response. And really the surprising result was here that only against Candida albicans we see this dominant Th17 production, but not against all these other fungi, so really suggesting that Candida albicans seems to be a major driver of this fungus-specific Th17 responses in humans. So, but then we also had a closer look again at the Aspergillus response. So we took these Aspergillus reactive T cells, we expanded them, and then re-challenged them again with different antigens. And as you can see here, they nicely re-react to Aspergillus, as they should. But interestingly, we saw that there's a small population of these cells that were initially stimulated with Aspergillus. It's also cross-recognizing antigens from Candida albicans. And when we now look again at cytokine production, we see after re-stimulation with Aspergillus, again, we have low IL-17, IL-2 production, so really mimicking this ex vivo response. But if we look at this small population of cross-reactive cells, so they produce really higher amounts of IL-17, IL-22, meaning that within this Aspergillus reactive response, there are some cells that also cross-recognize Candida antigens, and these cells obviously are the main producers of the Th17 cytokines, suggesting that actually these Th17 cells we find within the Aspergillus T-cell response may be induced via this cross-reactivity to Candida. We did not only find these cross-reactive cells in the Aspergillus response, but also in the response to a lot of other fungi, so it's indicating here. So we have a variable cross-reactivity also against these fungi to Candida. We never see any cross-reactivity to completely unrelated antigens like CMV, for example. And when we then again look at the cytokine production, we see that always these Candida cross-reactive cells, they tend to produce higher amounts of IL-17 and actually lower interferon gamma. So indicating that also in these responses, the Candida cross-reactive Th17 cells are present. Now, to further investigate whether this cross-reactivity really induces the Th17 response, we made the following experiment. So we again stimulated cells with Candida lysate, but then depleted the Candida reactive cells, so all the cross-reactive cells are gone, and then re-challenged the cells again with different fungi. And when we do this for Aspergillus, we see that the small population here of IL-17 producing cells is completely gone when the Candida cross-reactive cells are depleted, but for example, we do not see any difference in the interferon gamma production. And this is actually true for all the different fungi we analyzed so far. So in each case, when the Candida reactive cells are depleted beforehand, the IL-17 production is goes strongly down. So really suggesting that these Th17 cells we find against these different fungal species are mainly induced via this cross-reactivity to Candida. So what we think what's happening is that we actually have Candida, mainly a commensal in the gut, and this is obviously a potent inducer of these Th17 cells. But other fungi, like for example Aspergillus, obviously do not generate this type of response, but just by chance, some of these Th17 cells also recognize proteins from Aspergillus simply because they are similar or have a high homology. And by this T-cell cross-reactivity, we get an induction of these Th17 cells into the Aspergillus immune response. And so the next question was, of course, where does this play a role? So obviously, we all have these cross-reactive cells in us, so we find them in every donor, but we are all doing fine with that. But then, actually, we started to look also at the Aspergillus reactive cells in patients with pulmonary diseases, like asthma, COPD, and cystic fibrosis. And when we look in these patients, we see that, interestingly, this IL-17 producing cells are clearly increased in these patients, so mainly in the cystic fibrosis patients. And this is just the statistics to that, so we see this in the relative level within the reactive cells, but we also see clearly increase in the absolute cell numbers of Aspergillus-stimulated IL-17 producers in these patients. But interestingly, we do not find differences in the interferon gamma production, so suggesting that there's a selective expansion of these cross-reactive Th17 cells in the pulmonary diseases. Of course, this is all blood, so the question is always how relevant is this, because inflammation is locally maybe in the tissue, and in a great collaboration with the German Heart Center, we also had the chance to actually look in explanted lungs from cystic fibrosis and also control patients. And here we also could confirm that we see this increased IL-17 response against Aspergillus in the cystic fibrosis lungs. Again, no difference in the interferon gamma compartment. And just to prove that the cells that we look at are really derived from the lung and not from contaminating blood, we actually compared them to CD69 lung resident memory cells and also CD69 negative cells, which are more circulating cells. And here you can see that these Aspergillus reactive cells really mimic the phenotype of these lung-resident cells, so they express markers of tissue retention, but for example, downregulate markers of tissue egress. So really showing that the cells or that in the cystic fibrosis patients, we also find this increased Th17 response in the lung of the patients. So, but do they also contribute somehow to pathology in these patients? And to start to address this question a little bit, we further focus on this cystic fibrosis cohort. And these cystic fibrosis patients, they frequently are sensitized to Aspergillus, and 10% of them approx develop this ABPA, the allergic bronchopulmonary aspergillosis. And we could easily identify such patients that are actually sensitized to Aspergillus based on an increased Th2 cytokine production, and also which correlates nicely with an IgE response. And now when we look at these sensitized patients, we found that we find this increased Th17 response only in patients that have an acute ABPA. Interestingly, it rapidly goes down with treatment. So when patients are treated with itraconazole and corticosteroids, we do not find this in patients that receive chronic treatment due to recurrent ABPA, and also not in patients that had an ABPA history, but not at the time point of our measurement. And very interestingly, we do not see any differences in the Th2 compartment between these groups, so really showing that there's a selective increase in the IL-17 cells, and there was also no difference in other cytokines. So finally, to really show that also in these settings, these cells are cross-reactive, we again isolated the IL-17 producers and also the Th2 cells from the patients, expanded them and then re-challenged them. And as you can see here, the IL-17 producers react to Aspergillus, but they react really in the same way also to Candida antigens. So really showing that they are 100% cross-reactive. And interestingly, when we do the same for the Th2 compartment, the cells react to Aspergillus, but we see zero cross-reactivity, so indicating that these cells really recognize different antigens from Aspergillus that are obviously not present in Candida. So showing that these cross-reactive cells expand in pulmonary inflammation, and this strong association with the ABPA suggests that they may actively contribute to the manifestation or exacerbation of the ABPA. So to put this last part also in a model, so we think in the patients what we maybe have is an increased interaction with Aspergillus that somehow leads to this selective expansion of the cross-reactive Th17 cells. And especially when this comes together with a Th2 response, obviously it drives the pathology in these patients. And overall, I think our data really show that there's a critical role of fungal microbes for shaping the human immune system. So these Th17 cells we find against Candida are the strongest response we see of Th17 cells, also in a panel of other antigens. And of course, it raises the question of a systemic immune modulation by T-cell cross-reactivity against different microbes, because as humans, we do not live alone, but we have a lot of different microbes that somehow shape our immune system. And with this, I'd like to thank all people that contributed to this study. So most of the work was done when I was still a postdoc in the lab of Alexander Scheffold, before I recently started my own small group in Kiel. I really like to thank our collaborators from Jena, so Axel Brackhage and Olaf Kniemeyer, a really long-standing collaboration for everything we do with fungal pathogens, and recently also with Bernhard Hube and Sascha Brunke. So these are really the fungal specialists, so we are more the T-cell people here. And especially I also like to highlight our clinical collaborators, so without them we cannot measure any patient samples, and thank you very much for your attention.
Excellent. We're open for questions. Maybe I could again start. The very interesting work. Clearly there's something very special about Candida that distinguishes it from the filamentous fungi. What do you think some of the virulence factors are that contribute to the immunological findings?
Yeah, it's really an interesting question. So why Candida is able to generate this type of response? And I mean, all these different fungi, they have similar toll-like receptors and things like that. So we think that it might really be a question of the root, and also maybe of the doses, so we don't know exactly that. But what is known, for example, for Th17 cells from mouse models is that they arise in the gut, so in the intestine, and they need somehow an adhesion to the epithelium to generate this type of response. And this is something Candida is able to do, and we think this might be one reason why it's such a prominent inducer of the Th17 cells. For the filamentous fungi, I really cannot say. I just can say we do not find this in the human system, and also not in invasive infection. So it's not like in these conditions they suddenly start to do the Th17 response.
So Candida albicans, but there are other Candida species, make a particularly interesting virulence factor, candidiasis, which is a lytic peptide. I don't think the moulds have anything equivalent. Is something like that maybe part of the, the bias towards Th17?
Yeah, very good point. So the Candida lysine has been shown to directly damage the epithelial cells, and then they start also to secrete factors like IL-6, IL-1 beta, which are involved in Th17 priming. So this could be indeed one route how we get this Th17 induction by Candida.
Good.
My name is Esther Segal. I am from Tel Aviv University. My question is, you mentioned candidas, lysate, if I understood you right. Do you know or have any clue what in that lysate initiates that reaction?
Yeah, very good point. We are intensively trying to figure this out, which are the proteins that actually drive this Th17 response, and also proteins that drive the cross-reactivity. So we identified one pair, actually. So the Candida MP65 is a known protein of Candida, and when we made the homologous protein from Aspergillus, which is a cell wall glucanase, we can also see that these are directly cross-reactive and drive this Th17 response. But we are currently also further investigating actually which are the Th17 driving antigens from Candida, and we established method that we can elute the peptides again from the MHC molecules and then analyze them by mass spec. But these are things that are ongoing. So at the moment, I don't know yet which are the proteins that drive this response.
That's really interesting. Thank you. Very good talk.
I have a question. What do you think the linking factor is? The one disease activity data you showed was the acute ABPA expansion of the cross-reactive cells. Do you think that's directly related to the temporal fungal burden? Because that could be easily measured, whether you look at sputum galactomannan or perhaps even a PCR assay. And I wonder if there's any data in that regard.
Yeah, we have done this, so we have not directly correlated this because the clinicians always tell us when you take sputum from CF patients, it's full of everything. It's really difficult to directly quantify the Aspergillus in the
David
site.
could help with that answer, right?
Probably you have better methods to do that. But yeah, we think that really Aspergillus in this setting drives this expansion of these cross-reactive cells because it's a directly Aspergillus-related disease, and that's an interesting fact because you have this Th2 response that is present all the time, obviously, and just the addition of this Th17 response somehow drives then the development of the ABPA.
It does raise the question whether that's just an epiphenomenon or is actually driving some of the acute pathology, right?
Other questions? I wonder if I could maybe ask you one other question. I don't want to put you on the spot, and so maybe this is a question that the audience can participate in. So you did a lot of work on cystic fibrosis, a very, very interesting disorder. You know, at least in the United States, clinically, clinicians who deal with CF manage this disorder primarily with antibiotics, right? So inhaled tobramycin and you know, things directed at bacteria. What very uncommonly, is my understanding, are antifungals used, and yet your data add to the growing body of evidence that fungi are a very important part of the disease process. And so I wonder what do you think? Should we be more aggressive in treating the fungi that we frequently find in CF, and universal sensitization to Candida and other fungi being some of the evidence that they might be contributing to disease pathophysiology?
Yeah, difficult question. So I can just say from my point of view, when we started this project, exactly the idea was you have a CF patient that obviously has a problem, but it's not known, it's a bacterium, is it a fungi, and what else? And so we came into the field because we think that we can use the T cells actually as a diagnostic parameter to see what is going on. So do we see difference in the T cell response against Aspergillus or Candida or against Pseudomonas, Staph aureus, and so on? And we have very nice results on that we see somehow the cells behave different in different conditions, and we try to correlate this. And this could, in the end, lead to a more targeted therapy than doing more antifungals or more antibiotics. But we are not at this stage, right? So the question is what to do now, and so this question I clearly cannot answer because I'm not a clinician.
Sure.